Welcome, class. Today we are diving into one of the most important topics in biology, the history and discovery of cell. It's not just a chapter in your textbook, it's foundational knowledge you need for your WAEC, your JAM, and if you're a university student, I also recommend this class for you. So, we're going to cover key scientists and discoveries and how they built the modern cell theory. Now, I want you to pay close attention because these facts will appear in your exams. So let's begin. By the end of this lesson, you're supposed to know how to explain how cells were discovered, identify the key contributors to the cell theory, and state the three principles of modern cell theory verbatim. That's word for word. So let's begin. Why should we care about the history of cell? Three big reasons. The first, exam focus. You should know that WAEC and JAM love asking about scientists, their experiments, and cell theory principles. Second, it's foundational. You can't properly grasp genetics, medicine, or even today's biology without this background. And thirdly, practice links. This ties directly to microscopy, lab work, and breakthrough like stem cell therapy. So pay close attention because this is not just history, it's your key to acing questions and understanding modern science. Now, before microscopes, people had different ideas about life. For centuries, people believed in spontaneous generation, the idea that life could just when I mean spontaneous generation, I mean that the idea that life could just appear from non-living things like dust or rotten meat. But why didn't they discover cells earlier? Have you asked yourself this question? What the answer is because without magnification, cells were completely invisible. Now imagine that the entire building block of life is just hidden in plain sight. Now let's talk about the breakthrough that changed biology forever. The invention of the microscope in 1595, a Dutch eyeglass maker named Zacharias Janssen created the first compound microscope. This wasn't just a tool, it was like giving scientists superhuman vision for the first time. Before this, cells were completely invisible. After Janssen's invention, suddenly we could explore the hidden world of life. The single device paved way for every cell discovery that we are actually discussing today. Now, building on Jansen's microscope, an English scientist named Robert Hooke made the next big leap in 1665 when he sliced a cork very thinly and placed it under his microscope. He saw something revolutionary, tiny empty compartment that reminded him of monk's bones. So he called them cells. He published his findings, including detailed drawings in his famous book called Micrographia. But class, here is a critical YX jam point. Hooke only observed dead plant cells. That's what I want us to take note of. He only observed dead plant cells. The leftover structures after the living cells had died. He never saw living cells, and I want us to remember this distinction because this is a very possible exam question. So, so let's move over to Anton. So while Hooke saw dead cells, a Dutch trader named Anton made the next breakthrough using his handcrafted simple microscope, which could magnify up to 300 times. He became the first person to observe living cells in 1674, and he called them animalcules. Let's ask ourselves this question: what did he see? Bacteria from pond water, protozoo, even human sperm cells. He wrote that they moved very prettily, capturing the wonder of discovery. Now remember, WAEC and JAMB test this key distinction. When Hugh saw living cells, while Hooke only saw dead plant cells. I want us to take note of that. So I'm going to write it down now. So let's continue. Now let's fast forward to 1838. Two German scientists, Matthias Kleidin and Theodor Schwann, a zoologist and a botanist and a zoologist respectively, made the big connection. Sklein declared that all plants are made of cells, and a year later, Schwann confirmed that all animals are made of cells. Together, they laid the foundation for cell theory. But sharp class, note this: Wayek and Jam, for Wayek and Jam, both wrongly believed that cells could arise spontaneously from non-living matter. This error was later disproved, which we'll see in the next page. Still, you should know that their work was revolutionary. So, we're going to discuss about Rudolf Virchow now, the scientist who fixed a critical error in the cell theory in 1855. So, he declared in 1855, omnis cellula est cellula, which means all cells come from pre-existing cells. This simple Latin phrase demolished the old idea of spontaneous generation that Sclady and Stewart had wrongly accepted. Now, Virchow's principle became the final pillar of modern cell theory, and I want us to remember this. If Wayek and Jam ask who debunked spontaneous generation at the cellular level, now Virchow is your answer. Virchow is your answer. You must take note of that. So, let's talk about the modern cell theory. Built on Modern cell theory is actually built on the work of Hooke, Lewin Hooke, Skladin, and Schwann. And okay, I'll say that the most important person on this is Virchow. It has three non-negotiable principles, and I want us to listen carefully because Wayek needs this word for word. All organisms are made of one or more cells. The cell is the basic unit and structure of living things. And the third one is all cells come from pre-existing cells, thanks to Virchow. Notice how Principle 3 fixes Grading's and Schwann's error. That's why Virchow's contribution is non-negotiable for exams. I want us to write down these points because they are guaranteed to appear. We're going to move over to now. How did science finally bury spontaneous generation? Two landmark experiments proved Virchow's principle that life comes from life. First, it was Francesco's in 1668. Francesco Redi in 1668. What he did was that he placed meat in jars. Some of them were covered, some were open. And the result was that maggots only appeared in the open jars where flies could land. Simple, right? It's very simple, but that's a very powerful experiment. Then Louis Pasteur in 1859, he boiled broth in a special swan neck flask. Dust entered, but no microbes grew because germs got trapped in the curved neck. When he tilted it, life appeared. Pasteur's experiment was the final nail in spontaneous generation's coffin. And class, I want us to write this down. Pasteur's experiment is JAMB's favorite trick question. These centuries old discoveries are not just history, they feel today's biggest breakthroughs. Take stem cells, first isolated from human embryos in the year 1998. Yes, 1998. They are revolutionizing regenerative medicine, helping repair damaged organs. Then there is the CRISPR gene editing. That's what I call it. It's the CRISPR gene editing. So that's what some people call it, but I call it the CRISPR gene editing. That's 2012. It uses bacterial cell mechanisms to edit DNA-like genetic scissors, curing inherited diseases. And WAEC loves asking about the HeLa cells. Remember Henrietta Lacks, her immortal cancer cells harvested in 1951, accelerated drug testing and vaccines. Now, class, this is vital because cell theory's legacy is in every modern lab and hospital. And we shouldn't just call it a theory. We should never call it just theory. So, we're going to move over to some practice questions, and I want us to drop the answers in the comment section. So, hands down, who first observed living cells? Which principle did Virchow add to cell theory? And what did Hooke observe in Koch? So the answer to the first question is Anton van Leeuwenhoek. Remember his animicals. The answer to the second question is all cells from pre-existing cells. Now, notice how I said it. All cells from pre-existing cells. You have to take note of what I'm saying because you're going to be WAEC requires word for word. You should not add or remove any other thing. All cells from pre-existing cells. Omnis cellular excellular. And the last one, what did Hooke see in his Koch? What he saw was dead plant cell walls, not living cells. These exact questions appear in past questions, and I want us to revise it. Before we close, 3 battle-tested study strategies to conquer WAEC and JAMB cell theory questions. First, mnemonics. Remember this: Hooke, Leeuwenhoek, Schleiden, Schwann, Virchow. Just remember the letter H, L, S, S, V for chronological order. Now, second, diagrams. I want us to, before you should browse and search and sketch Pasteur's Schwann neck flask daily until you dream it. Examiners love labeling experiments. And my third advice for you is past papers. Solve at least 10 years of WAEC and JAMB questions on cell theory. Why? Patterns repeat. This topic is marks on platter. Revise HLSSV. You can do it this night. Draw the flask and go crush exams. I believe in you. You've got this. See you next time.